Hello, welcome to the ALS webinar. Please note that we will be recording the webinar, so please save your questions until the end. A recording of the webinar will be available to download shortly after the presentation. And today I will be presenting on arsenic speciation as a tool for monitoring exposure from food, drink, and occupation. Now, arsenic is a well-known poison dating back to ancient Egyptian times, but there are also some lessons to learn from our parents. So there was good reason for them to say, don't bite your nails, as this is where excess arsenic is stored in the body. Another good idea is to vary your diet so that food staples such as rice, bread, pasta are from a variety of sources, and I'll demonstrate why later. Bread and pasta as staples have also become sources of arsenic in our diet due to substitutes for gluten. So it's mainly the rice flours that are now introducing arsenic into what was traditionally didn't contain a food group that didn't contain arsenic. And on April 1st of this year, the US FDA announced an action level of 100 ppb of inorganic arsenic in rice and rice products. And the reason I mention inorganic arsenic is it's not total arsenic, it's just the inorganic forms. And these are typically high in products such as rice, also grapes and apples, so there may be action levels coming out from the US FDA for grapes and apples as well in the future, but that, to this date, hasn't happened. So ALS was fortunate enough to be involved in a project to decommission the Giant Mine roaster complex in Yellowknife, Canada. And the mine has a long and notorious and rich history. The Giant Mine was one of the richest gold mines in the history of Canada, with almost 10,000 ounces of gold produced in 1948 alone. So it produced 237,000 tons of arsenic trioxide, which is currently permafrosted in caverns under the mine, and is reported to be enough arsenic to kill everyone on our planet. So the project was interesting in the fact that workers needed to be tested before and after their shift to ensure that there had been no worker exposure from the mine site to arsenic while completing the project. Now, in the process of decommissioning the roaster complex, there was 2 million kilograms of arsenic trioxide removed. And in order to control the worker exposure, the consultant involved used a biological exposure index, which came from the ACGIH, Industrial Hygiene Group out of the US, of 35 micrograms per liter in urine. And this was used to monitor their exposure to make sure they're not being exposed to the arsenic trioxide that they were removing on site. Now, why this prevented many challenges, and why also it's essential that you use arsenic speciation. So you look at the difference between inorganic and organic species of arsenic, was because the source of arsenic was not just from the site, from the arsenic trioxide, but also from things that are very commonly eaten in the Yellowknife area. that include natural plants. In this particular case, we have in the top right-hand corner Labrador tea, which is a common tea drunk in that area. We also have cigarettes that are a source of arsenic. We also have a lovely picture here of the Northern Lights, but the Great Lake here, which is also a source of arsenic from drinking water and just water in general. We have the soil as well in the bottom left-hand corner. And then we have the food and diet. So in particular, the apple juice, the grape juice, and then the rice cereals and rice products and rice substitutes. And then also we have, in the bottom right, we have just natural seafood and the levels of organic arsenic, which are harmless, considered harmless or non-toxic. And these are in not just the fish, but they are also, in the case of sushi, you've got rice from the containing arsenic. You've got seaweed that also contains essentially a good form of arsenic, or arseno-sugars that are easily excreted from the body and not considered toxic. And then also you have the fish itself and shellfish that contains, again, a lot of organic arsenic, which is normally readily excreted from the body. So many challenges, because we're trying to monitor the urine that's just associated, or urine that is coming from worker exposure, from the middle picture there, which you see, which is the roaster complex that has been sealed up with plastic to contain not only the dust from arsenic trioxide dust, but also from any asbestos or anything that needed to be removed from the decommissioning of that site. So it's wrapped up like a giant ball left there. So in order to monitor the workers and to control the exposure, they decided to use this biological exposure index of 35 micrograms per liter. And the workers were monitored both before and after, so we took urine samples before and after. And if results were ever above the action level, which was just decided to be half of the actual exposure level of 35, so it's 17, then a very fast sample needed to be taken, urine sample. It was shipped down from Yellowknife to our lab in Burnaby in British Columbia, and we had to get that results back quickly. So that was just to make sure they're not getting towards this 35 micrograms per liter. And this is where diet plays a big part in this equation, because often the levels were above 17, and it was due to the worker eating fish before their shift, or rice before their shift, or had a cigarette before their shift, or whatever it may have been that had arsenic in it that put them over this level. But if monitored quickly afterwards, and also to look at the organic arsenic, you could determine that this would or would not be a problem. So in the case of a worker eating a lot of fish, it might have high arsenobutane, and this isn't inorganic arsenic and is not measured in the biological exposure index. So the way it works is that essentially the biological exposure index is looking at the inorganic arsenic species and the two major metabolites, which in this case are monomethyl arsenic acid, or MNA is short for, and dimethyl arsenic acid, which is DMA. So arsenobutane is not part of this, and it's considered organic arsenic, along with arsencholine and other arsenosugars that are harmless. So it's important to be able to distinguish between the inorganic arsenic that's more representative of exposure and the organic arsenic, which is often more representative of food, diet, and sort of lifestyle. So here's just a summary of the species that we were looking at and able to separate. The biological exposure index was based on the sum of the arsenate, which is arsenic 5, the arsenite, which is arsenic 3, MMA, monomethyl arsenate, and dimethyl arsenic acid, or DMA, and that was considered the sum. So that had to be below 35 micrograms per liter in the urine. And arsenobutane and arsenylcholine were also measured, and these were not part of that total, but they also were often responsible for why, if you were just looking at total arsenic and not using speciation, why you would be above this exposure index. So it's really critical that you speciate, because you don't want to add in this arsenobutane, which would be present if you weren't doing the speciation. And just as a little background here are some of the reference ranges of these compounds that you will see in urine. So you can see arsenobutane is often sort of in the sort of 10 ppb range in urine, and MMA is also present there. DMA is often the most common. It's the last stage of metabolism, so that is often common. And you see the arsenic, inorganic arsenic, 3 and 5 are often in the 1 to 2, 2 to 3 ppb range in urine. So in humans, the typical elimination pathway of inorganic arsenic, so 3 and 5, follows this scheme. It's 5, it's reduced down to 3, which is then further reduced to methyl arsenic acid, or MMA, and then it's further reduced to dimethyl arsenic acid, or DMA. So that's the typical pathway, and why you often see DMA in the urine samples of people, because it's actually your body metabolizing. And if it gets too much, the body can't metabolize it, and then you start seeing the inorganic arsenic showing up in the urine, and you also start seeing other indicators. It starts getting stored in the nails, starts getting stored in the hair, and then of course you start seeing, in some cases, toxic effects, cancers and other things that can form from your body not being able to remove the toxic forms of arsenic from the body. So the methods we actually use, there are lots of different techniques out there, but it was very key for this particular project, and also future projects where you have to analyze food, you have to analyze water, and also urine, is to have a robust method. And there are many techniques. There's atomic absorption, that was an older technique that was commonly used. This often would not allow the analysis of individual species, but would just give you a total arsenic. Then there was ICP-MS. Again, this could be coupled to other instruments to allow speciation, but it was often prone to polyatomic interferences, and in particular from the gases used to produce a plasma in the instrument in order to atomize the ions, and that would cause an interference. And so collision cell, reaction cell ICP-MS came along, which sort of took away a lot of these polyatomic interferences. And so ICP-MS, collision cell ICP-MS, really ended up being one of the instruments that was measuring better arsenic. But at this particular stage, we're still really looking at just total arsenic. And in order to look at the speciation, you've got to start coupling this instrument with other instruments. So in this particular case, you can couple them to, you can have hydride generation that's forming, and couple that to high-res mass specs, or you can couple it to, in this particular case, there's an HPLC coupled to a high-res ICP-MS collision cell and a quadrupole. They can all be coupled to various different instruments. This is a high-res instrument, which would be a very expensive way to do the speciation. But in the end, the technique that we went with was an IC or an HPLC. In our case, we chose an HPLC, which we have here. And the HPLC has been coupled to this collision cell ICP-MS. So this was the technique we decided to go with. And it was basically due to the fact that this removed a lot of the interferences that you often got. You still would get those interferences with the high res, which is to the right here, and also due to the just reliability and robustness of this instrument. So the HPLC allowed the speciation and separation of the species of arsenic, and then the collision cell ICP-MS was what was used to actually measure the arsenic that's coming through. So it ended up being, we've ended up following for the urine a US CDC method, and we were able to get very good sensitivity, way below the 1 microgram per liter range. So we could see natural levels of all of these species in urine. And in all cases, we were using the mass spec to determine the arsenic mass. And then we would always report the data as arsenic, so not as the concentration of the actual compound, but all as arsenic. And the advantage of using this HPLC coupled to an ICP-MS was that we also were able to detect arsenobutane and arsencholine, and these sometimes are often present in seafood and shellfish, and so it's a good indicator of whether the exposure has been related to the breakdown of those things from food, or whether it's related to worker exposure. So a typical chromatogram, which is how we are able to speciate. So this is, as it's coming off the HPLC column, you suddenly detect, in this particular case, the first one we see coming out is arsenobutane, then we have the DMA, then we have the arsenic 3, then we have the MMA, and then we have arsenic 5 coming out at the end of the column. So this is the sort of separation you get, and often you would have arsencholine here as well, and sometimes these both were present, and so it was sort of buried under this arsenobutane peak here. And that's just often due to seafood. You would see this large peak here. And it readily goes away when you see a couple of, 24 to 48 hours after you see this peak. You can often see that disappear as that food is processed and excreted from the body. So just looking at one of the many workers from this project over a period of 8 months, you can see that if you were to measure the total arsenic, and this is what these individual orange dots are showing, you can see that they often were above the action level. You can see all these points here that are above 17, but you can also see that a lot of these points are also above the 35 microgram per liter, which was also the biological exposure index, which would mean that the worker would have to change their duties on the site, or in some cases they would not be able to work on the site due to these levels being above the exposure, worker exposure level that the site has been managed to. So this 35, but you can see these points, and this is looking at total arsenic. And if we dive into a little bit more detail and look at our speciation, we can then see that if you separate these species, some of these, that one particular one there that we saw high worker exposure, a lot of it is related to arsenobutane and also DMA that may actually be present naturally in the seafood as well, So you've got this arsenobutane, which is putting it with 35 is a sort of biological exposure in it. So you can see in this particular case, it's the contribution of the arsenobutane that might prevent that worker from going on site and working, getting a day's pay. So in this particular case, again, arsenobutane, if you weren't measuring that, you wouldn't necessarily be able to control and look at the worker exposure. So in many of these cases, and this is we just took one example here of a worker, but those total arsenic values are not an issue when you look at the speciation. You can see that the total inorganic in many cases goes down below the 35 microgram biological exposure index that they're controlling the worker exposure on the site with. So that's looking at just concentrations of arsenic in urine. But I also wanted to just show you how using the same instrumentation, you can also see, if you're looking at, these are several rice flour samples, and for exactly the same reason, if you compare the inorganic and organic arsenic in rice flour, these are several different rice flours, the action level from the FDA is 100 ppb. Now you can see this organic arsenic is just tipping the rice flour over that 100 ppb limit. The blue is just inorganic arsenic. And then the red is just the additional contribution of organic arsenic. So if you did not speciate your sample, these particular samples would be flagged as above. So you've got this one here, this one here, this one here, and this one here would all be considered above the FDA 100 ppb action level. That's for rice flour. And in the case of rice grains, you see even more of a trend, where you can see, again, we've got the inorganic arsenic is the blue. You can see often even the inorganic is going over the 100, but often it's the contribution of the organic arsenic, which is putting, in this case, grains of rice and different grains of rice, it's just putting it over the 100 ppb action from the FDA. So you can see a lot of the cases, it's just the red that was doing it. But the most important point is to understand that you have to speciate. You have to use speciated arsenic and instrumentation that allows you to differentiate between inorganic and organic arsenic species in your samples. If you were just looking at total arsenic, there's going to be a lot of times where you are reporting levels that are above the FDA action level, and it's not representative of the toxicity of that arsenic. So in conclusion, total arsenic without speciation would certainly not provide adequate information to monitor worker exposure, and that's exposure to not only the workplace, but it's also exposure to water, food and drink, and even your lifestyle with cigarettes and cigarette smoke, which also contains significant amounts of arsenic. HPLC-ICP-CCMS, or high-speed collision cell mass spectrometry, that is the technique ALS uses, the instrumentation we use, as a sensitive and reliable and very rapid method to test for arsenic species in not only urine. We use that same instrumentation for water, food, and drink. And the reliability, it's always good to have several pieces of equipment. So again, because it's in this particular project, it was worker exposure and whether the worker was able to work on a shift or not. It's important to also have backup pieces of equipment so that you can maintain that. And so also we have several of these instruments so we can keep the throughput and keep the workers working on the site. So thank you. I hope you found that webinar interesting, and I'm open to take any questions.